from Shri Shivaji Vidya Mandir, Aundh. So today we will going to see that Griffith experiment. We have already know the DNA and what is the DNA. We already learned in a previous lecture as well as in a previous standard about the DNA. So basically this Griffith experiment are based on the genetic transformation. In Griffith's experiment there is one scientist named as Frederick Griffith. So that's why this experiment is called as a Griffith experiment. So in this experiment there are 4 type of strains. First is the rough strain, second is the smooth strain, third is the heat killed smooth strain, and fourth is the rough and heat killed smooth strain. Fourth strain is the combination of a rough strain and a heat killed smooth strain. So there are 2 types of a strain, the rough strain and a smooth strain. So basically smooth strain are virulent. So what you mean by virulent? Virulent is this is the some type of a poisonous. So second is the pathogenic. Pathogenic means because of this disease will cause. This is the smooth and encapsulated. That means this particular strain will covered with a capsule. And rough strain, it will be a non-pathogenic, non-virulent, non-capsulated. So it will be a S type and it will be a R type. That means smooth strain, rough strain. So in this experiment there is one bacteria are used named as a Streptococcus pneumoniae. So Streptococcus pneumoniae is the particular bacteria and which has a cocci shape. Cocci shape means small circle like shape, and this bacteria is used in this experiment. Because of this bacteria pneumonia is caused. Griffith first injected bacteria with rough strain to the mice. It will alive. That means mouse get alive. That means this particular strain doesn't affect on a mice. So second, he injected smooth strain to the mice and mouse get dead. That means this particular strain get affected and it causes pneumonia. So that's why mouse died. In third, he injected heat kill smooth strain to the mouse and mouse get alive. That means this heat kill smooth strain didn't affect on the mouse. And fourth is the rough strain. That means our first strain, rough strain, and heat kill smooth strain. That means this third strain. Combination of these strains injected and mouse get died. Pneumonia causes, that means mouse get affected by a pneumonia and get died. But first we have given rough strain to the mice. At that time mouse get alive, and when we injected heat killed smooth strain, at that time also mouse get alive. So how it can be possible? Both strains are giving a result which will be a mouse get alive, and when combination of these 2 strains given to the mouse, at that time mouse get died. So this Griffith concluded that there is some transformation of a strains which this rough strain get converted into or transfer into smooth strain. That's why this mouse get died. Because of smooth strain, mouse get died. So here also this rough strain get transferred into smooth strain, and this because of this smooth strain mouse get affected by pneumonia, and that's why mouse get died. So Griffith said that this change change was a genetic change. So Griffith said there is the transforming principle get used. That means that rough strain get converted or get transferred into smooth strain and bacteria get virulent, and that's why mouse died. Now we will see the second experiment, that is the Avery, McCarthy and MacLeod's experiment. They were a scientist and they proved that because of DNA, R strain get converted into S strain deadly. So in previous experiment we have seen that there is a transforming principle, and because of this principle R strain get transformed into S strain deadly. How it is happen? So we will see in this experiment. So in this experiment, this scientist first purified DNA, RNA and proteins and other materials from the cell, and after that it mixed with the R strain and heat kill smooth strain, but they mixed it separately because they want to see that which is the transformer. Basically they discovered with the help of a protein, RNA and DNA. So there are a protein digesting enzyme, DNA digesting enzyme and RNA digesting enzymes are present. So protein digesting enzymes are proteases, DNA digesting enzymes are DNAs and RNA digesting enzymes are RNAs. They mixes proteases in given strain. At that time mouse get died. After that they mixes RNAs in the given strain. At that time also mouse dead. That means protease and RNAs affected on the mouse and it causes pneumonia. That's why that particular mouse get died. After that they mixes DNAs in a given strain, and at that time mouse get alive. That means DNAs affect on mouse and because of this mouse get alive. That means here when protease and RNAs get mixed with strain and at that time mouse get died, then transformation didn't occur. But here when DNAs get mixed with strain, at that time transformation occurs. That means DNA is a transforming factor, and because of this DNA mouse get alive. So in previous experiment, that is Griffith's experiment, we have seen there is a transformation principle, and because of this transformation principle mouse get alive. So in next experiment we got to know that DNA is a transforming factor, and because of this DNA this mouse get alive. So transformation principle is a DNA. But all biologists didn't convinced. So that after this experiment some other scientists came and they proved that DNA is a transformation principle and DNA is a genetic material. So in a previous experiment we have seen that there is a something transformation principle is present, and that transformation principle is the DNA. But previous experiment doesn't prove that DNA is a transformation principle. So next experiment is the Hershey-Chase experiment, and these scientists were proved that DNA is a transformation material, and because of DNA that particular mice get alive. They show the experiment in that bacteria and viruses grows. How? We will see. First of all, there is a bacteriophage. Viruses which are infects the bacteria are called as a bacteriophage. So there are two types of bacteriophage. One is the sulfur label protein capsule and second is the phosphorus label DNA core. That means both bacteriophage has a sulfur and phosphorus. So they used a medium in that sulfur is present and in other medium there is a phosphorus is present. They use radioactive phosphorus. It is a 32P in the medium, and they used radioactive sulfur 32S. They grew some viruses on the medium, and that medium contains radioactive phosphorus and radioactive sulfur. So the viruses can be grown in the presence of a radioactive phosphorus which contains radioactive DNA. That means labeled DNA. But virus didn't grow on the sulfur labeled medium. Same as viruses grown on a radioactive sulfur which contains radioactive protein. But they didn't grow on radioactive DNA because DNA didn't contain sulfur. So the radioactive phages are probably allowed E. coli to grow on the medium, and this medium should contain phosphorus and sulfur. We have already seen that E. coli is a bacteria. Now you can see here this sulfur label protein capsule is present and phosphorus label DNA core is present. So this bacteriophage get infected in the medium. In next step you will see this label sulfur protein capsule and phosphorus label DNA core get enter into the medium, and this outer coating get removed with the help of a centrifugation. And in third step you can see only sulfur label protein capsule are present and phosphorus label DNA core are present. When this medium get observed, at that time there is sulfur is not detected in the cell, and here phosphorus didn't detected in the cell. We have already taken sulfur label protein capsule, and here we have already taken phosphorus label DNA core. So basically sulfur should present in the cell, but here didn't observe like that. Instead of present of sulfur, there is a absence of a sulfur in the cell, but sulfur is present in the suspendant. Bacteria which were infected by the particular virus which has a radioactive DNA. So that DNA were a material which passes from the virus to bacteria, and the bacteria which were infected by the particular virus which have radioactive sulfur, that indicates proteins from the virus didn't enter into the bacteria. That's why DNA is a genetic material which is passed from the virus to bacteria. So finally DNA is the transformation principle. That means DNA is a genetic material is proved. And finally Hershey and Chase experiment get all biologists, and hence the proof that DNA is a transformation principle. That means DNA is a genetic material.